Hi, in the next several videos we'll be studying photosynthesis, which is the process which plants and photosynthetic bacteria use to convert carbon dioxide into sugars and other high energy molecules such as ATP. The absorption of a photon from the sunlight creates a high energy electron, which can then undergo electron transport that powers the creation of a proton gradient, which drives the synthesis of ATP, just as was the case for oxidative phosphorylation. This is one of the few times we'll discuss plants in this class. Photosynthesis comprises two stages. The first stage are the light reactions. This is where sunlight is absorbed by chlorophyll molecules. High energy electrons are generated that drive the synthesis of ATP and NADPH. The dark reactions are referred to as the carbon fixation reactions, and carbon fixation is the process of incorporating inorganic carbon dioxide and producing organic hydrocarbons such as sugar and amino acids. These are independent of light but utilize the ATP and the NADPH that are produced by the light reactions. We will study both of these reactions in this class. Both of these stages occur in an organelle referred to as the chloroplast, which plants have, which has many similarities to mitochondria, which we'll see on the next page. Here are pictures of mitochondria and chloroplast. Plants also have mitochondria, but there are many similarities with chloroplasts. One of the key features of both these organelles are the double membrane structure, with chloroplasts having both an outer membrane and an inner membrane, and then they go even a third step and have thylakoid membranes. And it's actually the thylakoid membrane in chloroplasts is the analogous structure to the inner mitochondrial membranes. This is where the electron transport proteins occur. These are the membranes across which protons are pumped. This slide emphasizes the fact that both mitochondria and chloroplast have their own DNA and ribosomes, and we'll say more about that in class, as it's not particularly relevant to the energy generating process of photosynthesis. Here we see the protein machinery that is responsible for the photosynthesis process. All these proteins and chlorophyll molecules, which are these green squares or schematized, exist here in the thylakoid membrane. We'll go through each of these pieces in turn, describing their function. Let's say at first, though, that photosynthesis operates in general like a photovoltaic cell. The sunlight will enter into the center here, into a protein called the reaction center. And what it's going to do, it's going to transfer electrons from this special pair down to this side. So we'll have negative charges here, and we'll have an electron hole, or the absence of an electron here. So we're going to have a positive charge and a negative charge. So what we have is light-driven charge separation across the membrane. And so this, as you can tell, this is the generation of an energetic intermediate. Moving opposite charges further away from each other requires energy. It is energy that you could get back if you let those charges go. They would recombine, and because of their attraction to each other. So what the reaction center is going to try and do is that as it builds up electrons at this quinone and at this quinone, it's going to remove these electrons on these quinone molecules, like we saw in oxidative phosphorylation, before they can recombine. Then when you've taken these electrons away, you still have the positive charge here. This electron will be replenished ultimately by splitting water, taking and ripping electrons out of water molecules, leaving you with an entirely neutral reaction center again. Now remember that when a photon is absorbed by a molecule, it promotes an electron to a higher energetic state. Before that photon is absorbed, the electron is very happy to sit here. But the excited electronic state of these two chlorophyll combined is the electron is less stable on that than it is sitting on the neighbor chlorophyll to the left, so that the electron is transferred to this chlorophyll rather than staying in that excited state. This transfer occurs in a few picoseconds, which is faster than the amount of time that electrons in excited states normally return to their ground states of the molecule, which is about a nanosecond. So you've got about a 3 order of magnitude speed advantage for transferring the excited electron to the nearby chlorophyll molecule rather than returning to the ground state. Now I will erase some of these markings so that we can, because otherwise things will get too busy. Now, as I mentioned, the nomenclature is pretty awkward here. The main protein complex where the light generated charge separation occurs is the reaction center, and all these green squares are chlorophyll molecules, and there is one set of 2 chlorophyll molecules. It is a dimer. It's a pair of chlorophylls, and the actual name of those 2 chlorophylls is the special pair. I know it's rather confusing, but that's what it is. Surrounding the reaction center, we have antenna proteins, and these act just like antennas. If a photon had to hit just the special pair, not many photons would hit it that often. But what plants have evolved is antenna, lots of several hundred surrounding chlorophyll molecules, and these chlorophyll molecules are arranged in such a manner that they act as an energy funnel. The chlorophylls on the outer ring here have electrons that are slightly higher in energy than the lower ring, and the special pair has the lowest excited state energy of all of these, specifically because they're a dimer. So a photon that is absorbed by a chlorophyll molecule here will be transferred to other chlorophyll molecules. Now, this is different. This is not an electron transfer. It's not generating positive and negative charges. It's just generating the excited state. The photon is basically being transferred. This is a non-radiative process and is similar to the way that your radio antenna works. When you have an excited electron, it acts as an oscillating dipole, which is producing an electric field, and this electric field is vibrating, and that's stimulating electrons in nearby chlorophyll molecules, which then absorb that energy, and that energy is depleted from this chlorophyll, and it is transferred here via oscillating electric fields. So the photon will hop amongst these chlorophyll molecules until the photon hops on the special pair, and then you start to transfer electrons to another chlorophyll molecule, and then to the quinone, then to a second quinone, and then this quinone, when it gets 2 electrons on it, it will diffuse out into the thylakoid membrane. Again, these molecules are very hydrophobic. Even with the negative charge, they are not able to escape into the aqueous environment, and this quinone will transfer the energetic electrons to the electron transport chain in a manner exactly analogous to what we've seen for oxidative phosphorylation. So overall, this antenna system is very efficient, and it's why the chlorophyll is why the leaves are green, and a photon basically hitting any portion of the leaf surface will be captured and utilized for generating energetic electron. The last thing to mention is that since we're continuously pumping off electrons to the electron transport chain, we're constantly replenishing them from this protein that sits on top of the reaction center. And one of the amazing things that plants have evolved the ability to do is to take those electrons from water, which is a very low energy electron. Again, we in oxidative phosphorylation get our electrons from sugars, which we've oxidized. Here it's taking it from water, which would be which is an amazing engineering achievement. But this is all powered by the absorption of light energy, the process, the ability to extract the electrons from water molecules. And if it sounds like I'm a little excited, it's because I am. This is what I spent my PhD studying. I studied why the electrons, once they're excited, go left instead of right. There's a similar, almost identical mirror image pathway on the right, but the electrons do not go down that path. They always go to the left. So this is an energetic diagram of the photosynthetic process. I'll spend only a short amount of time of that on this slide, but on the next several slides we'll go through these together. The blue molecule is the special pair. The green molecule will consider the first quinone in the process. And the photon comes, it's going to promote the electron from the ground state to the excited state. Eventually, in a few picoseconds, the electron will be transferred to the quinone, well, microseconds to the quinone, generating a negative charge on the quinone, the positive charge on the thing, so you have light-driven charge separation, and then this electron will go off to the quinone. The special pair will be replenished from the protein that sits on top of it with a low energy electron. This is increasing the distance from the charge separation. And now the electron from water replenishes the protein sitting on top of the reaction center. Now here are both pictures on one slide. So we're starting off here, and then we have the absorption of a photon. The electron will be promoted here to an excited energetic state. We can draw it as a sort of larger orbital. And then the electron is transferred to the quinone. So we have the negative charge here and the positive charge here. And then the electron is donated back to this. We have the negative charge down here still. We have the negative charge making this neutral. And here we see the electron transport chain with a new few players. But here is the reaction center we've been talking about with the antenna around it. The electrons getting generated here go to the quinone molecule, and these will be transferred to the electron transport complex, the cytochrome B6F complex. Plants only have a single protein that's pumping protons rather than the three which we have in oxidative phosphorylation. And here we have the water splitting enzyme, which is splitting water, producing oxygen, protons, and electrons, which are being funneled into the protein. Now, what I haven't mentioned yet, but we'll see in the next video, is plants have two different types of light-absorbing photosystems. We have been talking about photosystem 2, continuing the trend of the poor nomenclature for this reaction center special pair. The whole process starts in photosystem 2. Later we'll get to photosystem 1, and we'll explain why there needs to be two photosystems at all. But a major point on this slide is that photosynthesis can also be called coupled vectorial processes, in that we have electron transport occurring laterally in the membrane. We have protons being pumped across the membrane, and we have proteins diffusing through an ATP synthase. I'll back up so you can see that. There's the ATP synthase. The protons are diffusing down their electrochemical gradient, generating the synthesis of ATP. And this will be a good place to stop. We'll resume our discussion on photosynthesis, talking about why there needs to be two photosystems. Thanks for listening.